Hi everyone, I am Dr. C.S. Kulkarni, MD, and I am going to teach medical biochemistry. Before I start the topic, please like, share, and if you are new to this channel, please subscribe it so that the next video will be intimated to you. In the last session, we ended up with functions of glycogen. In today's session, we will start with generation of CMP, in which we will see both liver and muscle are activated by cyclic AMP mediated activation cascade triggered by hormonal signal. Then we have protein kinase activation, where the cyclic AMP combines with the regulatory subunit so that the catalytic subunit is free to have its own activity. Then we have glycogen synthase, where both glycogen synthase and phosphorylase activities are reciprocally regulated. Lastly, we have glycogen storage disease type 1, which is nothing but 1-GK disease. Good morning, everyone. Let us start the topic. Generation of cyclic AMP. Both liver and muscle phosphorylases are activated by cyclic AMP mediated activation cascade triggered by the hormonal signal. The hormones epinephrine and glucagon can activate liver glycogen phosphorylase, but the glucagon has no effect on muscle. When the hormone binds to a specific receptor on the plasma membrane, the enzyme adenylyl cyclase is activated, which converts ATP to cyclic AMP. When level of cyclic AMP is high, it will activate a protein kinase. In this diagram, we can see that epinephrine and glucagon attached to a receptor on the cell membrane, which is bound to adenylyl cyclase, which converts ATP to cyclic AMP. This cyclic AMP converts inactive protein kinase to active protein kinase. This active protein kinase converts inactive phosphorylase kinase into active phosphorylase kinase. The protein kinase is inactive when the catalytic when the catalytic and regulatory units are associated with each other. The cyclic AMP combines with a regulatory subunit so that the catalytic subunit is free and to have its own catalytic activity. The protein kinase A is an enzyme that can phosphorylate serine and threonine residues of several enzyme proteins and is activated by cyclic AMP. This combines with the regulatory subunit of pKa. In the absence of CMP, the pKa is an inactive tetramer. R2C2 plus 4CMP gives 2C plus R2CAMP4. The intracellular concentration of CAMP therefore decides level of active pKa, that is protein kinase A, and cyclic AMP level depends on the activity of adenate cyclase and phosphodiesterase. Cyclic AMP level is increased by glucagon and decreased by insulin. Here we can see that ATP is converted to cyclic AMP with the help of enzyme adenylyl cyclase, and glucagon has a positive effect on adenylate cyclase. On the other hand, CAMP is converted into 5' AMP by the enzyme phosphodiesterase, and insulin has a positive effect on phosphodiesterase. The active protein kinase now convert the phosphorase kinase to an active phosphorylated form, which converts phosphorylase B to phosphorylase A. In this diagram, we can see epinephrine and glucagon in the liver and epinephrine in the muscle trigger the hormone receptor complex, which stimulates adenylyl cyclase. Adenylyl cyclase converts ATP to CAMP. On the other hand, the cyclic AMP is converted to 5' AMP with the help of enzyme phosphodiesterase. Here we can see cyclic AMP activates protein kinase A, which converts protein kinase inactive into protein phosphorylase kinase inactive to phosphorylase kinase active. This converts glycogen phosphorylase inactive to glycogen phosphorylase active. On the other hand, insulin activates protein phosphatase 1, which converts active glycogen phosphorylase to inactive glycogen phosphorylase. Coming to the next point, phosphorylase kinase activation. Phosphorylase kinase itself is a tetrameric enzyme, that is, it has alpha, beta, gamma, and delta subunits. Of the gamma subunit has of these, the gamma subunit has the catalytic site, and the other three subunits have a regulatory effect. The phosphorylase kinase is activated by calcium and phosphorylation of alpha and beta subunits by protein kinase A. Phosphorylation of alpha and beta subunits relieve autoinhibition of catalytic activity of gamma subunit. Binding of calcium to the delta subunit, which is the identical to calmodulin, is also necessary for full activity of delta subunit, since it also has the role in dysregulating the gamma subunit. Calcium triggers muscle contraction as well as glycogen breakdown through the action of phosphorylase kinase. The rate of glycogenolysis is linked to the rate of muscle contraction. The dephosphorylation of the active form of protein phosphate 1 involves removal of phosphate group from a phosphorylase A and alpha and beta subunits of phosphorylase kinase. The activity of protein phosphatase 1 is controlled differently in liver and the muscle. The catalytic subunit of protein phosphatase 1 in muscle is active only when it is bound to glycogen through glycogen binding subunit. The phosphorylation of PP1, that is protein phosphatase 1, by an active insulin stimulated protein A kinase activates the enzyme, whereas the phosphorylation at site 2 by protein kinase makes its action inactive. When cyclic AMP level is high, protein phosphatase 1 is inhibited by inhibitor 1, which is activated by phosphorylation by protein kinase A. The effect of cyclic AMP is not only by increasing the phosphorylation of enzymes but also by decreasing dephosphorylation. Coming to the next point, glycogen phosphorylase in liver and muscle. First, consider the glycogen phosphorylase in liver. The liver phosphorylase B is of inactive form. It becomes active on phosphorylation. The active enzyme is denoted as phosphorylase A. The enzyme is inhibited by ATP and glucose 6-phosphate. In the liver, the PP1 is regulated differently through the intermediary of glycogen binding site or GL subunit. The glycogen binding subunit complex binds to the R and T forms, that is relaxed and tense forms of phosphorylase A, but the phosphate group attached to the serine 14 is exposed only in tense state. PP1 can convert phosphorylase A to phosphorylase B. Glucose is an allosteric inhibitor of phosphorylase A. Insulin favors this effect by promoting the uptake and phosphorylation of glucose. Coming to glycogen phosphorylase in muscle. Skeletal muscle glycogen is degraded only when the demand for ATP is high. The regulation of glycogenolysis in skeletal muscle is by epinephrine. Glucose has No effect on glucagon has no effect on muscle glycogenolysis. AMP formed by degradation of ATP during muscle contraction is an allosteric activator of phosphorylase B. The active form of phosphorylase is referred to A, that is active and phosphorylated form, and the relative non-inactive phosphorylated form is B. On the other hand, the active glycogen synthase is dephosphorylated and the phosphorylated is relatively inactive. Glycogen phosphorylase is activated by phosphorylation by phosphorylase kinase that adds phosphate group to a specific serine residue of protein phosphorylase B. The phosphorylase kinase in turn is activated by protein kinase A or cyclic AMP dependent protein kinase. Phosphoprotein phosphate dephosphorylates by both phosphorylase kinase and phosphorylase B. Phosphorylase B is sensitive to allosteric effectors, but phosphorylase A is not sensitive. The high concentration of ATP and glucose 6 phosphate in a cell will inhibit phosphorylase B. The glycogen synthase and phosphorylase activities are reciprocally regulated. The same protein kinase which phosphorylates the phosphorylase kinase would also phosphorylate glycogen synthetase. The activity of the glycogen synthase is markedly decreased on phosphorylation. Insulin promotes glycogen synthesis by favoring dephosphorylation. Glycogen synthase is active in the dephosphorylated state. Phosphorylase kinase can phosphorylate glycogen synthase and inactivate the enzyme. For protein kinase A also can also inactivate the enzyme by the phosphorylation. In this diagram, we can see glycogen synthase A converted to glycogen synthase B, where pKa is stimulating enzyme. On the other hand, glycogen synthase B is converted into glycogen synthase A with the help of enzyme protein phosphatase 1. Calcium and calcium dependent CAM kinase also phosphorate the enzyme. The relative rates of glycogen synthesis and breakdown of their control by the action of pKa, that is protein kinase A, phosphorylase kinase, and protein phosphatase 1. The protein phosphatase 1 can activate glycogen synthase only after dephosphorylating and inactivating phosphorylase A. The regulation of phosphorylase and synthase is a typical example of multi-site phosphorylation for metabolic regulation. Control of allosteric effectors is superimposed on covalent modification. Glucose 6 phosphate can activate glycogen synthesis. Insulin activates protein phosphatase 1 and phosphodiesterase, which decrease calcium CAMP level and increase the G6P, or that is glucose 6 phosphate level by both to promote glycogen synthesis. The reciprocal regulation of glycogenolysis and glycogenesis is by covalent modification, that is phosphorylation and dephosphorylation. Insulin and glucagon are the major regulatory hormones, although epinephrine has stimulatory effect on glycogenolysis in both liver and muscle. They bring about alterations in the activity of protein kinases and phosphatases by varying the level of cyclic AMP. In this diagram, we can see the glycogenolysis. where glycogen N is converted to glycogen N minus 1 and Pi is converted to glucose 1 phosphate in the presence of enzyme phosphorylase, and epinephrine and glucagon are the have a positive effect on the enzyme. On the other hand, we have glycogen synthesis where glycogen N is converted into glycogen N plus 1 and UDP glucose into UDP and with the enzyme glycogen synthase, and insulin has a positive effect on glycogen synthase. Coming to glycogen storage disease type 1, it is also called von Jurke's disease. It is most common type of glycogen storage disease type, that is type 1. Incidence is 1 in 1 lakh live births. Glucose phosphatase is deficient in this case. Fasting hypoglycemia that does not respond to the stimulation by adrenaline. The glucose cannot be released from liver during overnight fasting. Hyperlipidemia, lactic acidosis, and ketosis are seen. Glucose 6 phosphate is accumulated, so it is channeled to HMP shunt pathway, producing more ribose and more nucleotides. Purines are then catabolized to uric acid, leading to hyperuricemia. Glycogen gets deposited in liver. Massive enlargement may lead to cirrhosis. Children usually die in early childhood. Treatment is to give small quantity of food at frequent intervals. This is the diagram of a child having von Jurke's disease, where there is short stature, delayed puberty, liver and kidney enlargement, fasting hypoglycemia, hyperlipidemia, and hyperuricemia. Here ends the session. Kindly like, share, and subscribe. Thank you.